Oh, wow, that was pretty spontaneous. Everybody just sort of quieted down. Well, welcome back. I hope everyone had a good reading week, despite, the traumatic event of the midterm. Whoops. All right, let's try this. And so, yeah, you saw the marks have been posted. I think we've worked out most of the issues with different anomalies on the midterm. So yeah, the makeup test will be held this Thursday, and after that, you'll be able to make an appointment, or you'll be able to make an appointment to come and view the midterm if that's what you want to do. So just watch for that. Dr. Dempsey will make an announcement about that. So just be patient, and he will get to that. Yeah, I'm not sure what else there is to say. It's a beautiful sunny day outside. So if you didn't do so well in the midterm and they're not as well as you wanted, it's good to be alive still. And on a nice day, a nice fall day like today, the sun is shining. I mean, there's always reasons to be thankful. Okay, so we are, those of you who were here at the end of the week before, realize we started into topic 14. So that lecture has been posted, the first part of topic 14. I'm going to finish off topic 14 today. So those of you who maybe haven't checked that out, you might want to check out the first part. This comic here, I like it because it does have the word biochemical in it, and it sort of underscores the idea that biochemistry really underlies absolutely everything, at least in Calvin's view. Even love is ultimately a biochemical reaction, which is something I like. Okay, so we're going to continue on. So I talked about DNA structure last time and stability of the double helix. I'm not going to review that, so that's in the previous lecture. But now I'm going to move on and just go over some differences between DNA and RNA, and I'll try to show that in pictures rather than words. The words are in the course notes, so you can look at that. So the first thing is, of course, DNA is based on deoxyribose, whereas RNA is based on ribose. And the difference is the OH group on the 2' carbon of the sugar in RNA is not there in DNA. So it's a deoxyribose there. That's one basic difference. Another basic difference is DNA will contain T thymine bases and not U. And the difference is the presence of the methyl group on thymine that is absent on uracil. RNA typically will have U instead of T. Okay, so that's another major difference. This is just going after the length. So point 3 is DNA is longer in general. It's a pretty important difference. RNA is copied from DNA and is only copied part of it. So by definition, an RNA is going to be shorter than DNA because it's only copying a small piece of the DNA. And then the fourth point here is that DNA, typically you get 2 complementary strands coming together to form a double helix, and that extends pretty much the entire length of the DNA, almost. Whereas RNA, you tend not to have complete complementary strands coming together. In fact, typically you'll have a single strand that is by itself without a complementary strand. And that leads to this sort of thing where the strand forms base pairing interactions within itself, right? So the strand itself will fold, so to speak, into distinct structures based on how the base pairs and the double helix can form within the single piece of RNA. And we'll see examples of that coming up. And then finally, base modification. So I gave the structures of the bases that you typically find in DNA and RNA, and you can find those anywhere in the textbook online. But there are certain modifications that happen to these. It's analogous to protein post-translational modifications like phosphorylation. You can add chemical groups to the bases in DNA and RNA as well, and this happens in both types of molecules. But the difference is DNA, there tends to be a relatively small number of specific modifications that get made, and they're not as common as they are in RNA. So RNA is a higher proportion of bases that get modified and are sort of deviating from the base structures that I presented. And so it's more common, and there's a larger number of different types of modifications. There's a lot greater variety in RNA than there is in DNA. So that's just, we're not going to worry about what exactly those modifications are or anything like that. I just want you to be prepared for that. So if you run into that later in your studies, you're like, oh yeah, McLaughlin mentioned that RNA gets modified, and this is one of those things. Okay. And so now we can look at an example of an RNA structure. And so the classic example then are the transfer RNAs, which are used to bring amino acids to the growing polypeptide chain, which Dr. Heinemann is going to talk about. So on the bottom here, it would be the primary sequence or the primary structure of this RNA. And even if just looking at that in itself, if you look closely, I know the text is fairly small, but you can see a number of differences between what you might expect and what's shown. So for example, we have D, which is not something I have actually introduced to you, but that's an example of a modified, in this case, a modified uracil. And there's other ones, like here's a Y, that's also weird. There's a psi, it's a Greek letter psi. And in fact, there's a T. And you wouldn't expect necessarily that. But the point then is it's going to that point of the modifications. It's not uncommon for bases to be modified in RNA, and there are examples, and you can look up exactly what these things are if you're curious. But then that primary sequence, of course, does not have a complementary strand that's can form a double helix with it. So the RNA tends to fold up on itself. And so this is what might be called the secondary structure map of the tRNA, when you can see that there are base pairs being formed between sort of self-complementary regions of the RNA molecule. And these are always occurring in an anti-parallel fashion, just the same way we saw that DNA is anti-parallel. But they're not, they don't extend the complete length of the sequence. You can see that double helix will form for a short stretch, and then it'll break, and then you'll make another double helix here. And there's in fact 4 major regions of self-complementarity in tRNAs, and the bottom 3 here form these so-called stem loop structures. So this, when you see this, you got a double helix, and then it breaks, and you've got a single-stranded region, and it comes back and loops on itself. We call this a stem loop structure. And you can name the loops as they've shown here. The anticodon, of course, is down here, which is used for translation. And if you look carefully, you can actually see some unusual base pairing, like there's a GU base pair that is allowed in RNA. Basically, the RNA molecule has a lot more flexibility in its structure that allows some what we'd call non-canonical base pairs, or non-Watson-Crick base pairing. So that most of the base pairs you see are Watson-Crick, so A base pairing with U and C and G, but you can get some non-canonical base pairs. And so that's what you might call the secondary structure map. And then this is a 3-dimensional representation of the tRNA structure. And you can see that if you look, you may be able to tell there's double helical regions here. So RNA can form double helices as well. The structure is not the same as bDNA. We're not going to worry about the differences between those, but it's the same idea. You get anti-parallel strands coming together that are complementary, that make base pairs, that have base stacking. It's the same idea. It's just the geometry and the structure is a bit different. But then because the fact that the double helices are not continuous, you can get some unusual structures forming. In this case, it forms sort of an L-type structure, which looks like a hair dryer or a power drill or something of that nature. And the anticodon loop is down here, okay? And so this is just to get the point that, you know, DNA structure is pretty regular, right? You got a bDNA double helix, and it pretty much forms more or less the same structure regardless of the sequence. Whereas RNA structure can vary a lot depending on the actual sequence and how that sequence causes these base pairing regions to form. Okay, that's the basic point. And as another example, this is the small subunit ribosomal RNA from this organism here. You will have to memorize this for the final exam. No, I'm kidding. Okay, so you don't have to memorize this structure. That would be, sorry. That's probably mean. It's probably mean to do that so soon after the midterm. You're probably still a little edgy. So don't at me, as they would say. Sorry, I should have done that. Anyhow, the point is, I'm showing you this, not so you have to memorize it, but to just give you the sense of how complex the RNA structures can be. So if you look closely, the base number 1 is right about here. And then you can follow the strand all the way, and the 3' end is down here. Okay, and there's over 1,500 bases in this molecule. But you can see then that where they come together, this is base pairing regions. You can see lots of stem loop structures. There's bulges, and there's different types of things that are going on here. And again, the point is not to memorize any of this, but just to give you a sense of this point here, that the RNA structure is sequence dependent, and because the way that this will fold up will depend a lot on where the complementary regions are. And there's a whole 3-dimensional structure, of course, that's not represented here, where these 2-dimensional things will fold up into a very complicated 3-dimensional structure. So RNA structure is much more like protein structure in this regard. Okay, so a protein structure is sequence dependent. What the 3-dimensional fold is depends on the sequence of the protein, as Dr. Bofa taught you. DNA is not like that, because not really, because you basically have the same more or less structure all the way through, no matter the sequence. But here RNA is back to that idea of sequence determining structure. Okay, and so that's that. Okay, so now that's just a brief tour of differences between DNA and RNA. And now we have to think about DNA packaging, because DNA is very long. We have about 3 billion base pairs in one copy of our genome. Okay, and if you do the math, that's 0.34 nanometers per base pair. It works out to about 1 meter of DNA. Okay, if you took all your chromosomes, laid them end to end, and stretch them out flat as a bDNA double helix, that's 1 meter, right, of DNA, and that's got to go into every single one of your cells. And you have trillions of cells, and the size of a cell, of course, is much, much smaller than that. Okay, and so there's got to be a way that we can squash or fit that DNA down into the size that it needs to be. So it's very long, but remember, it's very, very thin, right? So it's about 2 nanometers in diameter. So it's a very, very thin string, if you will, that has to be put into the nucleus. And so this here is just showing you nucleus, and the red is heterochromatin. That's chromatin that's really condensed, really as much as it can be, whereas euchromatin, which is most of the DNA here, is not as condensed as it could be, but because DNA has to be accessible. You can't just pack it in there. You've got to be able to get proteins in there to read the DNA to transcribe into RNA, and to even do repair, as we'll talk about. So the point is, we got to condense our DNA. So then how are we going to do that? So one idea that I thought you could use to condense DNA, I took from a gentleman in Minnesota, and he decided, he was a farmer, and he decided it would be a good idea to take some twine and roll it into a ball. And so he really got into this notion that this would be a good thing to do. So I'm not sure where he got all this twine. I don't know what kind of farms they have in Minnesota with all this twine, but anyway, this was in Darwin, Minnesota. And this is just a gigantic ball of twine that this gentleman decided to, make into a huge ball. And it's a nice sunny day here, but you might think in Minnesota it gets cold in the winter and so on. You might be worried about the twine ball, but they built it a little house so that it would be protected. Okay, and there's actually a museum here. You can go to Darwin, Minnesota, and in fact, there's a website in darwintwineball.com. You can check it out. But this thing, they say that this guy, Francis Johnson, in March of 1950, wrapped for 4 hours every day for 23 weeks. Now, I guess his farm, he had lots of farm hands that ran the farm or something while he was doing this, but you can see that it's a very big twine ball, weighs almost 8,000 kilograms. They don't know how long the twine is, right? Because he didn't think to keep track of that, but you can imagine it's a very, very long twine, and I'm sure he tied the ends together to make it longer. And I thought, well, maybe this would be a good way to condense your DNA. So let me ask you this. If you were thinking, and I haven't taught this yet, but what do you think is the best analogy for the most fundamental unit of packaging of a DNA double helix? So I got a few different options here. And so I'm asking for the most fundamental unit of packaging. Okay, I'll give you 10 more seconds. This is a different twine ball, by the way. This is a guy in Missouri who decided to make a twine ball. I'm not sure why that's such a desirable activity, but anyway. So a lot of people went for the telephone cord, but we have each other option is chosen, right? So it's a good question, right? If you've got this big long piece of DNA, how do you condense it down? Well, I would argue that the twine ball is probably not the best option because, as I mentioned, you need to access all parts of the DNA, especially if you've got genes in there, right? And once these gentlemen wrap these twine balls up, you're not going to get at the twine that's in the center of that thing without unraveling the whole thing, right? And so if you got sentimental or nostalgic and you wanted to see the piece of twine that started it all, he'd have to unwind the whole thing, and that's not very practical, okay? So we need some other way of packaging that will allow the DNA to be accessed by different elements, by different proteins. And I guess I should spend a moment too, just to define what I mean by chromatin, because we had like euchromatin here. What do we mean by chromatin? I didn't highlight this, but it's the DNA plus all the packaging proteins that are used to package the DNA. So there's a lot of associated proteins on the DNA, and so that plus the DNA, we call that chromatin. Some people pronounce it chromatin. It doesn't matter to me which way you pronounce it. But that's what we mean by chromatin. So there's got to be proteins that are organizing the DNA in some fashion. So those proteins in eukaryotes are called histones. These are the fundamental, and I'll answer the question. I haven't answered the question, but I've told you A is not right, but I haven't told you the answer, but we'll get there. Histones are the proteins that package DNA in their most fundamental unit. And there's different varieties of histones. The five main ones are shown here. Histone H1, H2A, 2B, H3, and H4. And there's different things you want to know about histones. They're fairly small proteins. They're not larger than 200 amino acids. They generally range from 100 to 200 amino acids in length, so they're pretty small. And they are rich in lysine and arginine residues. And that's what this table is trying to show you. If you take the amount of arginine and lysine, you just count them up in each one of these protein sequences, you get a range from about 1/5 to 1/3 of the protein is made up of arginine and lysine. If you take the average protein of any, just general proteins, it's only 11% are arginine and lysine. So that's what we mean. These are rich in lysine and arginine. There's a higher proportion of these residues in there. And if you're clever, you may have realized why this would be, because if you remember what Dr. Beaufort taught you, arginine and lysine are going to have positive charges at neutral pH. And the DNA has a sugar phosphate backbone. The phosphate is negatively charged. And so if you want proteins that are going to package and interact with DNA non-specifically, it's probably charge is a good way to go about that. And so these are highly positively charged proteins that are positive, so they can interact with a negatively charged DNA and package it. Okay, so that's why it's rich in lysine and arginine. And another thing I want to get across about these proteins is that they're highly conserved throughout evolution. And again, don't memorize this. That's another joke I could make. You have to memorize the sequence, but no, you don't have to. This is histone H4. It's the smallest histone. It's got 102 residues, and I'm showing the sequence in all of us, which is on the top line. Okay, and you can find histone H4 in all eukaryotes. All these histones is a fundamental packaging in all eukaryotes. Bacteria have different ways of packaging. We're not going to really talk about that. We're going to focus on you and me for now. But if we look at the different organisms listed here, this is Drosophila melanogaster. If you compare the sequence, you can line them up very nicely because there's only one difference. It's actually in the very N-terminal residue there. Otherwise, the sequences are identical. And you can go all the way down to yeast, Saccharomyces cerevisiae, and you see there's only 8 differences in the individual amino acid sequence between yeast, histone H4, and human. And if you think about the difference, right, between yeast and yourself, that's a huge evolutionary gap, right? And so, but these proteins are extremely well conserved. You're not going to find this level of conservation pretty much anywhere else, okay? What does that mean? So when we find residues that are conserved, and maybe Dr. Wolf has talked about this as well, they tend to be functionally important, right? So if you have residues in the active site of an enzyme, they tend to be conserved between organisms. Well, what this is telling me is that pretty much the whole protein histone H4 is important, okay? You don't want to deviate too much from the structure or the sequence of it, or it's going to have major problems for the cell. So all the histones are like this. They're very highly conserved throughout evolution, and that sort of reflects their fundamental importance in packaging of DNA. And if you get this wrong, you're probably not going to survive, okay? So if you have point mutations that mess this up, your DNA packaging is messed up, well, your organism may not survive that with that mutation. And if you look at the mutations, actually, it's interesting. A lot of them tend to be conservative, what we call conservative mutations. That means the properties of the different amino acids are similar, right? So we have arginine and lysine. So change from a lysine to an arginine is not that big. You know, a change from an alanine to a serine, well, that's just an OH group difference, right? That's why it's good to know the structures, right? Methionine to leucine, those are both hydrophobic residues, right? So the mutations, when they occur, tend to be conservative, which again underscores this idea that the structure of these proteins is very, very important. And so how do these then interact with DNA? So that's what we're getting to here. This is the nucleosome core particle. So what happens is you get 2 copies each of histones H2A, H2B, H3, and H4. So 8 proteins in general, in total, an octamer, and they come together and they form the structure here. And there's different colors, and each color is supposed to be a different histone. Don't worry so much about that. The point is that they come together in a cluster, and then the DNA wraps around the outside. And that's what these blue lines are. It's a double helix. And this is one representation. The proteins are in the ribbon structures here, and the DNA is in the wire. And if you take that and rotate it 90 degrees, basically the top coming down towards you, you get a structure that looks like this, where the dark blue is space filling model of the DNA, and the other colors are proteins, the histone proteins. And so, and it wraps around almost 2 times, the 1.7 times approximately, it wraps around the outside of the histone. And it, as a good Canadian, this sort of reminds me of a hockey puck. It's sort of that shape. It's a circular, squat little cylinder, which is the histone core particle. Or if you think about what the analogy I was talking about before, this would look a bit like a spool of thread, where you've got a central core of proteins around which DNA is being wrapped. So this is what we would call the most fundamental element of DNA packaging, the nucleosome core particle. Okay, and so it's, as I said, wraps around about 1.7 times. The crystal structure has 146 base pairs of DNA. Okay, and that is the fundamental unit. Now, the one thing you notice here, if you look at a couple of these proteins, they're pretty well structured in here, but then they've got these tails sticking out, right? So this yellow one, for example, goes way out here. The green one is coming out, and you can see that these are poking out the sides here. These histone tails, it turns out, become important in regulating DNA packaging, and it becomes important in expression of genes when you want to transcribe DNA into RNA. And Dr. Heinemann is going to talk more about this, but the main point I want to make now is when you wrap DNA around the histone core particle here, you're inhibiting RNA polymerase from getting at it. So the packaging of DNA into these nucleosome core particles tends to inhibit transcription. Okay, and so, but that's important for gene regulation. If you want to transcribe the gene, you need some way of reorganizing the packaging so that RNA polymerase can access the DNA. And Dr. Heinemann is going to talk more about that. I'm just priming you with that, those ideas, so you'll be ready when she picks up that strand and starts teaching you more about that. Okay. So those are nucleosome core particles, the most fundamental unit of packaging. And so you get the DNA wrapping around almost 2 times, and then this happens repeatedly. So if you take DNA from a cell and you treat it the right way, you can sort of disrupt all the higher order packaging, and you can get what we call this beads on a string image, where we have, this is an electron micrograph, and it gives you some sense of scale here. But we've got these clumps, these little dots or circles here. Those are the nucleosome core particles, and you can see then they're joined by a little line. That's the DNA. Okay, so the DNA goes around one core particle, and then there's a linker region, and then it wraps around another histone octamer, and then there's a linker region wraps around another histone octamer. So the whole DNA is decorated, so to speak, by these histone proteins, these histone octamers, with the DNA wrapped around. And the linker regions are not always the same length, and so I've given you approximate, range here from about 3 to 80. That's typical. Okay, you can't go much below 3, but you could have longer than 80 in principle, right? So get what's going on here. We've got the nucleosome core particles linked by these linkers. And so strictly speaking, the technical definition then of a nucleosome is a nucleosome core particle plus one of the adjacent linkers. So that's how you, strictly speaking, define a nucleosome. However, in common usage, the word nucleosome is often used to refer to the nucleosome core particles. Okay. So, and I expect when Dr. Heinemann comes, she'll refer to this as a nucleosome, because that's what pretty much, that's the way the word is used. But strictly speaking, if we want to be technical, the other DNA does exist. So a nucleosome consists of a core particle plus an adjacent linker. Okay, so that's just a sort of a technical definition. But when people say nucleosome, they pretty much mean this. Okay. All right. So now comes to an interesting point I want to make. Just pay attention because the final exam's coming. How big is your typical nucleosome? So if you're stopped on the street, or if you're sitting in a room with other people with your head hunched over a desk at some point in the near future, and it comes to your mind that you need to know how many base pairs are in a typical nucleosome, a good answer would be about 200, okay? Because you've got 146 base pairs wrapped around the nucleosome core particle, or the histone octamer itself, and the nucleosome core particle. And then the approximate size of the linker, let's call it about 50. Okay, so if the linker is about 50, you add 146 to 50, you get a number that's approximately 200. Okay, so if someone stops you on the street and asks you that question, now you'll have a good answer. Okay, I don't know how much more plain I could say. You want to know that number. About 200 base pairs per nucleosome. Okay, on average. Of course, individual nucleosomes are going to vary. Okay, so that's the most basic fundamental unit, but that is not going to get us condensed all the way down to what we need. This condenses by about a factor of 3. So what's next? Well, now we have these so-called 30 nanometer chromatin fibers, and that's what happens when the nucleosome core particles are packed together into a more regular structure, repeating structure. And this then requires histone H1. You'll notice histone H1 was not involved in the nucleosome core particle, but it is an essential histone. You need it. And what it does, and I borrowed this figure from a different book, but it binds to the nucleosome core particle at the spot where the DNA is emerging, and leaving the core particle, right, where the linker is starting. And if you get this histone H1 binding at all the nucleosome core particles, what you end up generating is a regular structure. And this is a experimentally derived image of that, and this is sort of a cartoon that's trying to represent what is going on in the top image. And they've numbered the nucleosome core particles, N1, N2, and so on. And so and the DNA is green here. So you can see the DNA is wrapped around one, and like it looks like a hockey puck, just like I said. And then it sort of goes, when it leaves that, it goes across to N2, and then it wraps around that, and then it comes across to N3, and wraps around that, and then it goes to N4. And so it sort of goes back and forth, and then you get these nucleosome core particles are stacked on each other, and it almost looks like a double helix itself, except this is a left-handed double helix, and you can verify that yourself later or right now, that they're sort of coiling around each other in a left-handed fashion. But this then would form your 30 nanometer chromatin fiber, so-called because the diameter of the thing is about 30 nanometers. Okay. So now you can see this is now working to really shrink down the length of the DNA by wrapping it and going back and forth like this into these fibers. And this requires histone H1. Okay. If you don't have histone H1, these will not form in such a nice way. Okay, and that's basically all we need to know there. And then 30 nanometer chromatin fibers can go into larger loops and other forms of packaging. Here it gets a little more vague. I mean, when we talk about nucleosome core particle, we know exactly, we got the crystal structure, we know exactly what it looks like, and then we can see these. And here we've got this to work with, and we can make a model, so we have a sense. But now the 30 nanometer chromatin fibers, they wrap into bigger loops, and it becomes less regular. And like they're showing regular loops here in this diagram, I think for simplicity's sake, because artists like symmetry and stuff like that. But in fact, you get these loops, what you might call that figure 8 sort of thing, and they can vary in size from about 30,000 to 200,000 base pairs. Okay, so the loops are not always the same size, and they are anchored to other proteins that are not histones, and that involve some RNA. We're not going to go into the identities proteins. People are still arguing about which proteins are in there, but we call that the nuclear matrix. This is different from the mitochondrial matrix, right? So the nuclear matrix is simply referring to a complex of RNA and proteins that form some sort of a scaffold or some sort of a structure to which the loops can bind, and then they extend, and then they come back and bind. It's sort of an organizing structure or scaffold. And am I being vague? Yes, I am, because a lot of these, the structure is going to vary from one place to another. It's going to be different. So basically just get the idea. 30 nanometer chromatin fibers will bind to this scaffold and then loop off and then come back and then loop off and some other structure and come back. Okay, so it gets a little less regular at this point. But the basic idea then is this: then we put all these things together, and it allows the DNA to be condensed in a way that will fit inside the nucleus, but still allows for DNA to be accessible, especially if you can change the positioning of these nucleosome core particles as needed. Okay, and Dr. Heinemann will talk more about that. Okay, so that's as far as I'm going to go there with packaging. Okay, and that takes us to the end of topic 14. We talked about the basic structure of nucleic acids. We talked about BDNA being a right-handed antiparallel double helix, and I talked about how it was stabilized. This lecture, I talked about RNA and the weird structures it forms, and then we talked about chromatin and different levels of packaging of DNA. Okay, that takes us to the end of topic 14. So now topic 15 is about DNA replication. We need to make copies, right, of the DNA. And so here we are, we're now driving towards the final exam. So we've covered the basics of nucleic acid structure. Today I'm going to talk about replication. I'll finish that next time, and then we'll go into DNA repair. And I should say my last lecture will be Thursday, okay? And then Dr. Heinemann will take over next week. And that'll be a good thing for you. So here's topic 15. We're going to talk about different stages of DNA replication: initiation, priming, and synthesis, and proofreading, and ligation. We're going to talk about definitions, what's going on in a replication fork and bubble, what telomeres are, and why they're important to think about. And I'm going to just briefly talk about inhibitors of replication at the end. Now, again, I know you've seen DNA replication before. This concept should not be new. You take a double-stranded DNA, and you want to make 2 copies, right? You want to end up with daughter strands, so to speak, parent strands and daughter strands. And this is required if you're going to divide your cells, right? Because each cell needs a copy of the genetic information. right? Each one of us started out as a single cell, and your magnificent self has got trillions of cells in it, and that's all because of DNA replication happening many, many, many times. And so this process is known as semi-conservative, and the idea is that each parental strand is used as a template to make a new complementary strand, right? So, and then you end up then with two daughter strands that hopefully, if everything has gone well, are identical in sequence to the original parental DNA. So here they're showing the two strands in different colors, blue and red. And here they're showing a thicker strand and a thinner strand. The point is that's what we mean by semi-conservative. Half of the DNA in the parental strand was the one original template, and the other half is completely newly synthesized DNA. And Watson and Crick, figured this out, or they predicted this would be the way it was copied, just based on their structure, because it's really handy to have the two complementary strands, because you have the sequence of one strand and you automatically know what the other sequence should be. So that makes it very easy to imagine how you would synthesize this by taking one strand and just putting a complementary strand next to it. Okay, so that's semi-conservative, although that still had to be proven experimentally. So that's what these folks did. Meselson and Stahl demonstrated that DNA replication was semi-conservative. The textbook describes it briefly. It's been called the most beautiful experiment in biology. You can look it up if you like. I'm not going to go through it. But I did want to just mention Meselson and Stahl. This is a picture of Meselson and Stahl along with someone else they knew, Martha Chase. Now, Martha Chase was involved in the famous Hershey-Chase experiment, which finally demonstrated once and for all that DNA was the genetic carrier, the carrier of the genetic information, because as late as 1952, this was still in some dispute. And so the Hershey-Chase experiment sort of pretty much nailed that down. And then Meselson and Stahl did this experiment to figure out that it's semi-conservative. Now, and I showed this photo, one, because I think it's nice, they're sort of relaxing in a sunny day. But I also want to call attention to the fact that they're young. These people were all young. They're very famous for the experiments they did, but they did them all before they reached the age of 30. So they're all in their 20s when they did all these experiments. And that just goes to show you that just because you're young doesn't mean you can't do great things, right? So just think about that. There's a very wise man once said to one of his proteges, don't let anyone look down on you because you're young. So I encourage you to take the same approach. Now, be sensible. You can't just go off and do weird stuff and say, well, I'm young, I can do whatever I want. But just because you're young doesn't mean you can't, do things that will be memorable and great things that can leave a mark. So have belief in yourself. And so that's just the thing there. So Meselson and Stahl, look it up. It's a great experiment. Don't have time to talk about it. So these are the steps in DNA replication. And we're going to go through them sort of one at a time, but this is sort of organize you. We got initiation, we got priming, we got synthesis, which includes proofreading. It happens at the same time. And then we've got ligation when things are sort of fixed at the end. Okay, so we're going to go through these different stages. Initiation, this is a very good place to start, you would think. That's when you start DNA replication. And DNA replication starts at specific sequences called replication origins. And so it doesn't just happen anywhere in the DNA. There are specific sites where DNA replication will start. And if you're a bacterium like E. coli, you have a fairly small genome. E. coli has only a single origin of replication. And so replication always starts at the same sequence, and that's well identified. And it turns out that bacterial replication origins pretty much look like each other. So we can use sequence similarities. If we find a new species of bacterium, we can guess where the replication origin is based on knowledge of other bacterial replication origins. So that's interesting to know. It turns out that in us, we have much larger genome than E. coli. And furthermore, we have linear chromosomes as opposed to one circular chromosome like E. coli. So we need multiple replication origins, many. In fact, 30 to 50,000 replication origins in a human being. That's a lot. And it's a lot harder to find them in eukaryotes. It turns out that the sequence of replication origins is not so well defined in eukaryotes. So it's harder just to tell from the sequence where they are. And in fact, the best definition for a replication origin is where the initiator proteins bind. Okay, that's the best definition we have. we look to see where the initiator proteins bind, and we say, oh, that must be a replication origin, because we can't tell just based on the sequence. There are certain similarities that they have. They tend to be AT rich. And if you remember, or if you know about DNA stability, AT rich sequences tend to be easier to pull apart than GC rich sequences. So that makes some degree of sense. But replication origins, they don't look identical in eukaryotes. Okay, so you can't, it's hard to tell where they are. But in bacteria, we definitely can tell. Either way, in different type of organisms, the basic idea is the same. We have special initiator proteins, and we're not going to go into detail about what those are, how they work. It's a little too much detail for us. What we're just going to do is say there's initiator proteins that they bind at the replication origins, and at the right time, they open it up. And they separate the two strands at the replication origin. And that leads to this structure here, where you've got double-stranded DNA flanking these single-stranded regions. We would call this is the beginnings of a replication bubble. The replication bubble is where you've got the two strands pulled apart. And then where the single-stranded regions meet the double-stranded region, we call that a replication fork, because the DNA is separating. It's like a fork in the road, right? And this is where helicase is going to be active. And helicase is an enzyme that is designed, or that has the function of separating double-stranded DNA and extending the replication fork in either direction away from the origin of replication. Okay, so we got a replication bubble, we get strands separating, helicase will bind there and make the replication bubble bigger by moving in either direction away from the replication origin. So that's a function of helicase. And this reminds me then, this idea of helicase reminds me of the great philosopher Yogi Berra, who unfortunately did not win the Nobel Prize, but he did win 3 American League MVP awards, which, if you ask me, is probably more impressive, because Nobel Prizes, they give up to 3 people, and there's 6 categories a year. So 18 people in the world could win a Nobel Prize in any given year. There's only 2 people who win the Major League Baseball MVP awards, and he won it 3 times. So that's pretty, I don't know, I'm a baseball fan, so this is a pretty big accomplishment. But anyway, Yogi Berra is known for his different sayings that he would make, which make you scratch your head. But the one that I'm thinking of now is advice to young people. And he said, when you come to a fork in the road, take it. I don't know what that means, but if you can figure that out, then go ahead. But come to a fork in the road, take it. Well, that's what helicase does. Helicase comes to the replication fork and it takes it. Okay, so it binds at the replication fork. Sorry, I go to great lengths just to have a little joke. So there's helicase binding at the replication fork. And then you have other proteins that bind, and they're called single-stranded binding proteins. And these bind to the single-stranded DNA and prevent it from reannealing, prevent it from forming double-stranded DNA again, because we want that DNA to remain single-stranded so it can be copied to make a complementary strand, so we can get 2 double helices in the end, right? So initiation will involve initiator proteins, which I'm not really describing in detail. Helicase will be there, and its job is to extend the replication bubble. And then single-strand binding proteins bind to the single-stranded DNA to keep it single-stranded, so that the action of helicase is not undone, so to speak, by reannealing. Okay, so that's initiation. So now let's imagine we've got a single strand here we want to use as a template for synthesis. And you might think, okay, there's DNA polymerase is going to come along and start synthesizing DNA, a complementary copy. It turns out DNA polymerase is unable to do this. If you give DNA polymerase everything you think it should need, it won't proceed. It will just sit there and not make new DNA. It turns out that DNA polymerase needs an existing 3' end to add things to. So we have a special enzyme called primase, and its job is to make a short, and by short I mean 10 to 20 nucleotides, a little short piece of complementary RNA on the template strand. So we have a different enzyme, not the regular DNA polymerase. This is a primase, and it'll take DNA and make RNA, just a very short thing. And it'll go in the 5' to 3' direction. It'll start at the 5' end of a new strand, and it'll make more or less complementary. Primases are not that accurate, okay? They make some mistakes because energetically it's hard to do this, which is why DNA polymerase doesn't actually do it. So primase, though, it can approximate the sequence of the DNA there, or complementary sequence to the DNA. And then it will dissociate, and that leaves a 3' end that now DNA polymerase can work with. So once primase does its thing, it dissociates, and DNA polymerase will bind, and it will be able to extend, and now it'll synthesize a complementary copy of DNA using a template strand as the template. And so this DNA polymerase, the structure of it is sort of shown here, at least the core of it, and it's got this interesting structure where you've got the fingers, thumb, and palm, and the DNA will sit in the middle of this hand sort of structure, and the active site is down there somewhere, and it'll extend the DNA from the 3' end. And this DNA polymerases were predicted, right, by Watson and Crick, but then people had to go find them. And so Arthur Kornberg was the first person in the world to isolate and purify and prove and characterize a DNA polymerase. And so for that, he won the Nobel Prize in 1959. He's looking pretty smug there. So this all happened in pretty rapid succession, right? 1952, it was finally proven that DNA was genetic material. Watson and Crick, the structure of double helix in 53, and by Nobel Prize in 59 means he discovered it before that. Moses and Stahl did their experiments in 58. It's interesting though, his lab discovered the first DNA polymerase, and they thought, okay, now we know how DNA gets copied. But then they did some math, because math is sometimes important, and they figured out that the DNA polymerase he discovered was too slow in E. coli to make a new copy of the DNA. Because in order for it to split or divide at the rate that it does. So they gave him the prize, and they said, wait a minute, he didn't actually find the thing that must copy the DNA. But he did find the first DNA polymerase. So it turns out his son, which one? Thomas, his son, found the DNA polymerase that's involved in replication. So bailing his old man out. He found DNA polymerases 2 and 3, and this 3 is the one that actually does replication in E. coli. It turns out Arthur Kornberg also had another son named Roger Kornberg, who did a lot of work with RNA polymerases and won the Nobel Prize in 2006. So that's a father-son Nobel Prize team. And then Kornberg had a third son who was an architect. So, 2 out of 3 ain't bad. No, nothing wrong with being an architect. It's fine. It's just not a biochemist like his other 2 sons. Anyhow, so Kornberg found DNA polymerase 1, and it has this basic structure. But the point is that any DNA polymerase works more or less the same. It's just DNA polymerase 1 is too slow. DNA polymerase 3 will do this. It'll take, any of them will take the 3' end of an existing double helix, an existing strand, and it'll use the other strand to put complementary bases all along, right? Using the Watson-Crick base pairing scheme. So anytime it sees a C, it'll put a G, and so on. And therefore you get a complementary strand. So that's basically that. The reaction itself is shown here. And you get here's a deoxynucleotide. You get a deoxy NTP that has 3 phosphates on it. And here's the 3' end, right? And I underscore this. It's always 5' to 3'. What we mean by that is the new bases are added to the 3' end of the growing strand. Here's the free OH on the 3', and the enzyme will catalyze this reaction where the OH attacks this alpha phosphate here, and you'll lose pyrophosphate to create a new phosphodiester bond, and then you've got a new 3' end, which then can undergo the same reaction again, and then you get a new 3' end, and so on and so on. So when we say synthesis is 5' to 3', we are talking about synthesis of a new strand. The bases are added to the 3' end. Okay, that's what we mean. And you can see then, if you remember what I told you about ATP, that this should be an energetically favorable reaction. This process of hydrolyzing phosphoanhydride bonds is energetically favorable. So the substrate for the reaction brings the energy in that allows the reaction to happen, which is pretty clever. Okay, so that is the basic reaction of DNA synthesis, and I'll probably pick up on this slide again on Wednesday.